Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk a little bit about gene regulation. Every cell in a multicellular organism contains exactly the same DNA. Even though this is true, we know organisms contain cells that can look very different from each other and have very different functions. This is only possible because of what we call selective gene expression. This essentially means that only certain genes are expressed in certain cells. A different set of genes is expressed in a different set of cells, and a different set of gene is expressed in other types of cells. These same genes may be inactive in different cell types within the same organism. Scientists have worked for years to determine how the processes of selective gene expression and gene regulation are carried out in organisms. We're going to start by talking about how it works in prokaryotes, bacteria, since these are the simplest case possible. Bacteria have these structures called operons. What an operon is this. It's essentially a set of related structural genes and the other nearby DNA sequences that help to regulate the expression of these genes. So again, an operon is a set of related genes called structural genes and the other parts of the DNA around that help to regulate the expression of those genes. We might think of an operon as a functional unit of transcription and gene regulation. For a gene to be on or expressed, it's got to be transcribed and translated. That's what it means for a gene to be on. It has to be making proteins. Operons usually consist of several parts. We're going to talk about those parts from a picture. So let's go to the next slide and let's look at a picture. So let's start off by talking about the structural genes. These are the actual genes that are making proteins from the operon. So those are the genes. The other parts of this operon help to regulate the expression of the genes. So there's a section of the DNA called the promoter. This is the binding site for RNA polymerase, the enzyme that carries out transcription. In between the promoter and the structural genes is a segment of DNA called the operator. You might think of the operator almost like the on-off switch for a gene. In some situations, the operator is going to have one of these things here represented in blue called a repressor protein attached to it. If a repressor protein is blocked to the operator, the gene is off. What that means is because RNA polymerase binds at the promoter, if there's a repressor protein at the operator, the RNA polymerase can't make it to the structural genes. And since it can't get there, those genes aren't transcribed, therefore they're not translated, and therefore the genes are off. Now, on the other hand, if there's no repressor protein bound to the operator, RNA polymerase will go straight through. It will transcribe those genes, use it to make RNA, and then the RNA will be translated and proteins will get made. In that case, the genes are on. At another place in the DNA, not necessarily adjacent to the promoter, the operator, and the structural genes, is another gene called a regulatory gene or regulator gene. This is the gene that actually codes for the repressor protein. So we're going to talk about a couple of kinds of operons. We'll first talk about inducible operons. In an inducible operons, the structural genes are usually not expressed. The genes are usually off. What this means is that the repressor proteins are usually attached to the operator, and the transcription of the structural genes is therefore blocked, and therefore those genes are not expressed. But under certain conditions, either environmental or maybe internal, a substance called an inducer will attach to that repressor protein. Think back to cell signaling. When things attach to proteins, it causes conformational changes in the proteins. So once that inducer attaches to that repressor protein, it changes shape. And this causes it to let go, to unattach from that operator. And once it's not bound to the operator anymore, RNA polymerase will transcribe the structural genes. They'll get translated and expressed. The most commonly cited example of an inducible operon that you hear about in AP biology is the lac operon. The structural genes in this operon code for a group of enzymes that are collectively referred to as lactase. So these are the enzymes that break down the sugar lactose. So here's the lac operon. And again, remember, it's an inducible operon. Normally, there is an active repressor bound to that operator, and therefore the genes are not expressed. But what will cause them to be expressed is this. Let's say that you've got some bacteria that find themselves in milk. Well, because they're in milk, there's a lot of lactose. And it wouldn't make sense to let that lactose go to waste. Without lactase enzymes, the bacteria can't metabolize the lactose. So lactose, when it's present in those cells, binds to the repressor proteins, as you can see here, causing them to change shape, fall off of the operator, unattached from the operator. And once that happens, the structural genes in here are transcribed, so they're used as a template for making RNA. And then the RNA is translated. It's used to make enzymes. In this case, these enzymes are lactase. And these enzymes then allow the bacteria to break down, metabolize, and use the lactose for energy. Once all that lactose is used up, the piece that's attached to the repressors also lets go. That means these repressors are now active again. So they'll reattach to the operator, and once again, the genes will be off. They won't be expressed anymore. So this is very similar to like a negative feedback loop. So again, let's go back and kind of refresh our memories from what we just said. So if bacterial cells don't have access to lactose, it doesn't make any sense for them to make lactase. So when there's no lactose present in the environment of the bacteria, the structural genes of the lac operon, they're not expressed. If, however, the bacterial cells get access to lactose, the lactose itself acts as the inducer. It binds to the repressor proteins, changes their shape, causing them to let go. This allows the structural genes to be transcribed and translated. The cell now has lactase. It can metabolize and use the lactose as a food source. Once it's all used up, the lactose, the repressor proteins, the lactose that's bound to the repressor lets go. The repressor proteins once again bind to the operator, and the expressions of those lactase genes ceases. And again, this is much like a negative feedback loop. It allows prokaryotes to express genes only when they need to be expressed. It's allowing the expression of genes to be regulated by the environment. This is a way to help to save energy and resources. It's crucial for the survival of the bacteria. Now, there's also another kind of operon called a repressible operon. The structural genes in repressible operons are almost always expressed. They're usually on. This means that the repressor proteins are not normally bound to the operator. Under certain conditions, either environmental or internal, a substance known as a co-repressor can bind to the repressor protein and change its shape so that it now will bind to the operator. This blocks transcription and translation and ultimately the expression of the structural genes, and essentially turns those genes off. The most commonly cited example in AP biology of a repressible operon is the trip operon. The structural genes in this operon code for a group of enzymes that can manufacture the amino acid tryptophan. This amino acid is essential for bacteria, and they don't normally get it from their diet or from their environment, so they have to make it. And these enzymes, coded for by the trip operon, allow the bacteria to make the amino acid tryptophan. So here's the trip operon. Again, normally, since it's a repressible operon, there's no repressor bound to the operator. RNA polymerase will transcribe the genes, so they'll make mRNA. The mRNA will get translated. We'll get the enzymes, the proteins, and those proteins will manufacture tryptophan, which the bacteria have to survive. Now, if the bacteria find themselves in an environment where there's lots of tryptophan present, it doesn't make sense for them to make something that's already there. So the tryptophan itself acts as a co-repressor. Here's tryptophan. It binds to the repressor, which was just floating around inactive before, changes its shape, so that now the repressor and the tryptophan together bind to the operator. They block transcription, block translation, and essentially turn off the expression of the structural genes of the trip operon. That's going to be the case until the tryptophan in the environment gets used up. Once it's used up, this tryptophan that was attached to the repressor is also used. Once that happens, the repressor protein goes back to its inactive shape, and the genes, the structural genes, will once again be expressed. So again, this works very much like a negative feedback loop. It's a way for bacteria to control the expression of their genes in relation to changes in their environment. It's very important for controlling and conserving resources. So that's how prokaryotes regulate the expression of their genes. In eukaryotes, it's way more complicated, and we're still learning about the way eukaryotes regulate gene expression. But we're going to talk about how they can do it on several different levels, and we'll start off by talking about chromatin structure. If you think back to Unit 5, you learned that eukaryotic DNA is usually not in the form of a chromosome. It's usually sort of unwound in a form called chromatin. You also learned that DNA in eukaryotes is often associated with proteins known as histones. These histone proteins help to organize, compact, and regulate the expression of the DNA. DNA is often organized into nucleosomes, pieces of DNA wrapped around groups of histones. And the degree to which the DNA is compacted and coiled around those histones affects the accessibility of the DNA to RNA polymerase. And thus it affects whether the DNA can be transcribed, translated, and ultimately expressed. Active genes, genes that are being expressed, are typically only loosely compacted and coiled around the histones. In that form, the DNA is called euchromatin. So that's active, expressed DNA. Inactive genes, on the other hand, are tightly compacted and tightly coiled around the histones, and those pieces of DNA are referred to as heterochromatin. Genes can be inactivated when methyl groups, CH3 groups, are attached to the cytosines of a gene. The additions of these methyl groups or methyl tags causes the DNA to tightly wind around the histones and therefore become inactive or not expressed. In most cases, methylation is permanent. Methylation deactivates specific genes in specific cells and causes different kinds of cells to differentiate from each other. So this is how in the human body, even though we have the same DNA in every cell, cells in your stomach look very different than cells in, say, your skin. And cells in your skin look very different than, say, cells in your brain. This differentiation, this deactivation of certain genes causes cells to differentiate. Another example of methylation is the bar body found in mammalian females. We know that each cell in a female mammal has 2 X chromosomes, but it turns out only one of those is active. The second X is highly methylated, and this causes it to compact into a tiny little ball-like structure called the bar body. And the genes on this X are completely inactive and essentially produce no proteins. Another kind of tag that can affect the chromatin structure is an acetyl group, a COCH3 group. Acetyl groups often attach to the histones themselves. This causes the DNA that's wrapped around the histone to uncoil, and thus the genes on that DNA are transcribed and expressed. Acetylation, the process of adding these acetyl groups, is very important in allowing cells to respond to changes in their internal and or external environments. Acetylation is usually reversible. We said that methylation is usually permanent. Acetylation is usually reversible, and it's important, again, in responses to changes in the environment, either internal or external, not so important in terms of differentiation. So here we can see methylation happening. So here's DNA that's methylated. So the methylated DNA is tightly wound around the histones, and then the unmethylated DNA is not nearly as tightly wound. This can be transcribed, whereas this is inaccessible to the RNA polymerase. Here we see acetylation. So here the DNA is fairly tightly wound up top, But once the histones become acetylated, and you can see these ACs, these are the acetyl groups, they attach to the histones themselves and cause the DNA to unwind and become accessible to RNA polymerase. So there's a whole new field of genetics called epigenetics. Epigenetics refers to changes in the expression of genes that don't depend on the nucleotide sequence itself. Acetylation and methylation are often linked to this field of epigenetics. Epigenetics is responsible for the changes that occur through the years in identical twins. We know that identical twins have identical nucleotide sequences, but as they get older, they look more and more different. That's not due to changes in their nucleotide sequences. It's due to changes in the methylation and acetylation patterns in their genomes. Because they're living maybe in different places, they have different lifestyles, they live in different environments, the methyl tags and the acetyl tags that are added to their DNA become more and more different across time. Turns out that methylation and acetylation patterns can also sometimes be inherited from the patterns. In most cases, these epigenetic tags are removed from the DNA during either meiosis or right after fertilization. But it seems there's a few cases where the methylation and acetylation patterns are actually transmitted from the parent to the child. Scientists don't understand how important yet this is in humans, but it is possible that the diet, the lifestyle habits, and the exposure to certain toxins in the environment of your parents can actually be passed on and affect changes in the child's DNA and your DNA. And that's kind of scary. But we're learning more and more about epigenetics. It's a intense field of research right now. So let's also talk about a couple more ways in which genes can be regulated in eukaryotes. In addition to changing chromatin structure, eukaryotes can also use what we call transcriptional controls. In eukaryotic cells, there are these proteins called transcription factors that help to regulate transcription. They bind to RNA polymerase, or they assist the binding of RNA polymerase to the promoter. So they sort of turn on transcription. Most eukaryotic genes are also regulated by transcription activators that can enhance transcription or repressors that block it. And these can change based on the internal environment. There's also post-transcriptional control. So once the pre-RNA is made or a primary transcript is made, it has to be processed. One of the most important parts of RNA processing is the removal of the intron sequences, the non-coding sequences, and the splicing together of the exon sequences. We talked in the previous unit how the RNA can be alternatively processed or alternatively spliced. And this allows one gene to actually code for multiple proteins. Here we can see that happening. So we have 6 exons in the original primary transcript of RNA. So that's the primary transcript. This is the actual DNA. So here's the primary transcript RNA. And notice that primary transcript has both the exons and the introns in light blue. So these are all introns that I'm circling. Depending on what's going on in the body, the internal environment, that primary transcript can be spliced in different ways. Here, notice all 6 exons end up in the mature mRNA. And once that's translated, we're going to get this specific protein. Here, though, notice that exons 2 and 4 are left out. So we get a different protein. Here, only exon 2 is left out. So we get even a third protein. So because of this process of alternative processing or alternative splicing, one gene can actually code for multiple different proteins. And that increases our diversity of proteins that are available. This is important in the way we make antibodies, the way we make receptors to certain things like smell. This allows us to have only a few genes for antibodies, only a few genes for smell receptors, but ultimately end up with lots of different antibodies and lots of different smell receptors. All right, so that's it for different ways in which organisms, both prokaryotes and eukaryotes, can control the regulation and ultimately the expression of their genes. Hope that helps.